Hi students, welcome to Smart Locus. In this video, we will see the chapter Photosynthesis in higher plants, second part, that is dark reaction and factors that influence the rate of photosynthesis. First about dark reaction, that is biosynthetic phase of photosynthesis. It is known as dark reaction. Light reaction means it takes place in presence of light. But dark reaction, actually the name is a misnomer. The word meaning it takes place in darkness, but it is not like that. Actually it indirectly depends on light reaction. It depends on the products of light reaction, that is ATP and NADPH. Immediately after light becomes unavailable, dark reaction continues for some time, then it stops. So, the name dark reaction is a misnomer actually. Melvin Calvin conducted experiment by using radioactive isotope of carbon, that is C14, in algal photosynthesis. That is an algae named Chlorella pyrenoidosa. Then, in this experiment, he found that the first stable product of carbon dioxide fixation is a 3-carbon compound, and it is named as PGA, phosphoglyceric acid. Later, other scientists discovered that in some other plants, the first stable product is not 3-carbon compound, instead it is a 4-carbon compound. So it is concluded that carbon dioxide assimilation is of two types. In some plants, PGA, 3-carbon compound, is the first product. We call it as C3 pathway. In some other plants, oxaloacetic acid, a 4-carbon compound, is the first stable product. We call it as C4 pathway. Calvin cycle or C3 cycle. C3 cycle is the biosynthetic pathway. That is the pathway that yields glucose in all photosynthetic plants. Doesn't matter whether they have C3 pathway or C4 pathway. Now let us see the steps in C3 pathway. There are 3 steps in C3 pathway. That is carboxylation, then reduction, then regeneration of RUBP. These are the 3 steps. Let's see the first step, carboxylation. It is the most crucial step of C3 pathway or Calvin cycle, where carbon dioxide is fixed or carbon dioxide fixation takes place, or RUBP is carboxylated to form a 3-carbon compound, that is 3-phosphoglyceric acid. And this carboxylation is catalyzed by the enzyme RUBP carboxylase. Since this enzyme has oxygenase activity also, it is better called as Rubisco, that is RUBP carboxylase oxygenase. It is a bifunctional enzyme. Usually enzymes are highly specific, but this enzyme has 2 functions, both carboxylase activity and oxygenase activity. So this is the first step, formation of 3-phosphoglyceric acids. Then second step, reduction. Reduction step utilizes, actually it involves a series of reactions, ultimately forms glucose. So reduction step involves a series of reactions that ultimately yields glucose, which will be stored as starch in plants. And this reduction step utilizes ATP, energy, and NADPH. ATP for phosphorylation, NADPH for reduction. Here, 2 ATP and 2 NADPH are required per carbon dioxide molecule fixed. That is the requirement of reduction step. Then, next, third step, regeneration. Regeneration of RUBP is also crucial. That is to continue the pathway uninterrupted. And this step utilizes 1 molecule of ATP for phosphorylation. 6 turns of Calvin cycle are required to produce 1 glucose. A single turn yields only 1 carbon, but glucose has 6 carbon in it. Therefore, 6 turns are required to produce 1 molecule of glucose. Single turn of the cycle requires or utilizes 3 ATP, 2 for reduction, 1 for regeneration of RUBP, and 2 NADPH for reduction. Therefore, 6 turns require 6 into 3, 18 ATP, 6 into 2, 12 NADPH. So 18 ATP and 12 NADPH are required to synthesize one molecule of glucose in C3 pathway. About Rubisco enzyme. As I said earlier, Rubisco is a bifunctional enzyme. It has binding site for both oxygen and carbon dioxide. It is actually the relative concentration of oxygen and carbon dioxide determines which of the two will bind on the enzyme. And it has greater affinity for carbon dioxide. But in C3 plants, some oxygen also binds on this enzyme, Rubisco. As a result, its carboxylating activity gets decreased. When oxygen binds to Rubisco, RUBP, instead of converting into two molecules of 3-PGA, it will be converted into phosphoglycolate, a 2-carbon compound, and 3-PGA, 3-carbon compound. See, this is catalyzed by the enzyme Rubisco. Here Rubisco shows oxygenase activity. This pathway is known as photorespiration. In this pathway, there is no synthesis of ATP or sugar. And it involves or it results in release of carbon dioxide. That means fixed carbon will be lost in the form of carbon dioxide with the utilization of ATP. And the biological function of this pathway is not known yet. Then, this pathway does not occur in C4 plant. Photorespiration does not take place in C4 plants because they have a mechanism that increase the concentration of carbon dioxide at enzyme site. That means site of Rubisco. When C4 acid is transported from mesophyll to bundle sheath, within bundle sheath, this C4 acid, it undergoes decarboxylation which releases carbon dioxide. That increases carbon dioxide concentration in bundle sheath cells, minimizing. This activity minimizes the oxygenase activity of Rubisco. Promote carboxylase activity of Rubisco. C4 pathway. So those plants in which the first stable product is a 4-carbon compound are known as C4 plants. First of all, C4 plants are adapted to dry tropical climates. And their features includes, they have a special leaf anatomy known as Kranz anatomy, and they tolerate high temperature. They show response to high light intensities, and they lack photorespiration, and they have greater productivity of biomass. Kranz anatomy. Kranz means wreath. It is the reflection of the arrangement of cells. Bundle sheath cells are actually large cells seen around vascular bundles. They form several layers around vascular bundle. And these cells are characterized by large number of chloroplasts. And they have thick cell wall. They have no intercellular space. And they have agranal chloroplast. That is chloroplast without grana. Without grana means photolysis will not take place. Non-cyclic electron transport will not take place. That means there is no oxygen evolution here. And Rubisco enzyme is present in this chloroplast. That means this Rubisco will never come in contact with oxygen. So in C4 plants or in Kranz anatomy, mesophyll has normal chloroplast. Normal chloroplast. Here, the carboxylating enzyme is PEP carboxylase, phosphoenol pyruvate carboxylase, which is highly substrate specific. That means it fixes only carbon dioxide, catalyzes only carboxylation. Bundle sheath cells has agranal chloroplast, where the enzyme Rubisco is present. And the agranal chloroplast, the evolutionary purpose is to avoid oxygen evolution here. And since the bundle sheath cells are thick-walled and closely packed, oxygen cannot, it will not allow gaseous exchange. So oxygen cannot enter. Then how carbon dioxide enters? That is explained by C4 pathway, a cyclic process, also known as Hatch and Slack pathway. Actually, that is the primary objective or function of C4 pathway, to transport carbon dioxide from mesophyll to bundle sheath. C4 pathway, Hatch and Slack pathway. Here, at first, in mesophyll, PEP, phosphoenol pyruvate, is the carbon dioxide acceptor. That is a 3-carbon compound. Accepts carbon dioxide and forms oxaloacetic acid. That is fixation. Oxaloacetic acid is the first stable product. Then, which will then be converted into malic acid, another 4-carbon compound, which will then transported through plasmodesmata to bundle sheath, where this malic acid undergoes decarboxylation to form a C3 acid, that is pyruvic acid. During this decarboxylation, the released carbon dioxide is accepted by RUBP. It enters into Calvin cycle to produce glucose. So, say glucose is produced in bundle sheath chloroplast by Rubisco enzyme. Then, this pyruvic acid then transported back through plasmodesmata to mesophyll. Pyruvic acid will be utilized for the regeneration of phosphoenol pyruvate. During this regeneration, 2 ATP are required per molecule. That is, to regenerate 1 molecule of PEP, 2 ATP are required. So, see here, single turn of this C4 cycle yields 1 carbon dioxide in bundle sheath cells. 6 turns of C4 cycle are required to release 6 CO2 to synthesize 1 molecule of glucose. How many ATP and how many NADPH would be required for the synthesis of 1 molecule of glucose in C4 plants, not in C4 pathway? C4 pathway, there is no synthesis of glucose. In C4 plants, how many ATP and how many NADPH would be required? Total 30 ATP are required. How? 18 in C3 pathway, 12 in C4 pathway. Here 2 ATP per molecule. So 6 turn, 6 into 2, 12. 12 plus 18, 30. So 30 ATP are required. Then how many NADPH? 12 NADPH only, that is in C3 pathway. So 30 ATP and 12 NADPH are required to synthesize 1 molecule of glucose in C4 plants. Factors that influences the rate of photosynthesis are of 2 types: internal factors or plant factors and external factors or environmental factors. Internal factors includes number, size, age, orientation of leaves, mesophyll, chloroplast. Then, internal carbon dioxide concentration, amount of chlorophyll. External factors includes availability of sunlight, then temperature, then carbon dioxide concentration, then water. Then, Blackman's law of limiting factor. That is, if a chemical process is influenced by many factors, then the rate of the process is determined by the factor which is nearest to its minimal value, present in least quantity. Light. There is a linear relationship between incident light and the rate of photosynthesis at low light intensities. But at higher light intensities, a rate does not show further increase because other factors becomes limiting. And a very interesting point here is light saturation attains at about 10% of the total sunlight. As a result, light rarely becomes a limiting factor for photosynthesis. Next, about carbon dioxide concentration. Atmospheric carbon dioxide concentration is very low, that is 0.03 to 0.04%. Increase in this carbon dioxide concentration up to 0.05% increases the rate of photosynthesis. At low light conditions, neither C3 nor C4 responds to high carbon dioxide concentration. But at high light conditions, both C3 and C4 plants show increase in the rate of carbon dioxide fixation. C4 plants, carbon dioxide saturation attains at about 360 ml per liter. That is 360 ml carbon dioxide in 1 liter air. Whereas in C3 plants, C4 360, C3 450 milliliter per liter. That means when we increase the concentration of carbon dioxide, C3 plants are benefited more. Temperature. Plants require optimum temperature for photosynthesis. Tropical plants have higher temperature optimum than temperate plants. Similarly, C4 plants have higher temperature optimum than C3 plants. C4 plants require optimum temperature in the range of 30 to 40 degrees Celsius. C3 plants, it is in the range of 20 to 25 degrees Celsius. Then water. Availability of water affects the rate of photosynthesis indirectly. That is, water stress causes closing of the stomata, which will decrease the absorption, that will reduce the absorption of carbon dioxide. Then, water stress causes wilting of leaves, which will decrease the surface area of leaves and metabolic activity as well.